Hello my friends, I'm back. This time with an analysis and information that may one day affect you or somebody in your family directly. The issue is the current state of the art in treatments for Alzheimer's disease and dementia. Before I come to the point, allow me to introduce myself to those who do not know me. My name is BERNT von Grabe. I am an analyst since I'm out of my diapers. In the mid to late 60s, I was researching and teaching at the Frankfurt, Germany Institute for Planungstechnik, a privately funded think tank. My subjects were intelligence research, information technology, and business process analysis. In the late 60s, I showed that the human intelligence, measured as IQ, is not a fixed measure but dependent on exposure to intellectually challenging stimuli. In the mid-80s, Siegfried Lehr and Robert Langer showed that the absence of such stimuli caused a loss of IQ. Use it or lose it. It took the scientific community over 30 years to accept my findings, which today are the standard. In the late 80s, I discovered a process to regenerate partial liver resects to full size in vitro. In the mid-90s, I engaged in Alzheimer's research and came up with a pharmaceutical formulation, ADT2, that worked exceptionally well and restored the cognitive functions of test subjects. The effect unfortunately diminished after a while due to the acquisition of tolerance by the test subject to one component of the drug. We worked that out in ADT3. So, let me come to the issue today. In 1991, Dr. John Hardy, researching in the UK, postulated that beta amyloid depositions in the brain was the root cause of a pathologic cascade that resulted in Alzheimer's disease. Dr. Hardy is now an investigator at the National Institute of Health. Based on John Hardy's postulate, the pharmaceutical industry invested billions in developing a drug that targets amyloids. PFIZER, WTETH, Eli Lilly, ELAN, you name it. PFIZER alone offered $750 million for the rights to a drug and actually paid $250 million upfront for a product, DIMEBON or DIMEBON, I'm not sure I pronounced it properly, that only worked in Russia. What is the incentive to invest such large sums? Well, that is easy. In 2010, an estimated 35.6 million people worldwide lived with dementia. That figure is expected to rise sharply to 65.7 million in 2030 and 115 million in 2050. In other words, the Alzheimer segment of the population will quadruple between now and 2050. So, what did the pharma research get us so far? I hate to tell you this, but the big pharma suffered one dismal failure after another. First, PFIZER, the drug that allegedly worked in Russia and that targets amyloids, failed. Next, WTETH and ELAN. According to a report, their drug targeted amyloids and failed. Next, NEUROCHEM, a Canadian pharmaceutical company, their drug failed. Next, Eli Lilly, the drug that targets amyloids, failed miserably. Indeed, Lilly's failed drug raised the question whether the Hardys postulate that amyloids are the cause of Alzheimer's is valid. So, let's look into the question whether the amyloid theory holds any water. Enters Mark Smith, a researcher at Case Western Reserve University. Dr. Smith suggests that rather than causing Alzheimer's, amyloids are the result of it. Let's look for a second at the correlation between the degree of dementia and the amount of amyloids in patients. Indeed, there is none. Post-mortem biopsies have shown that patients who were severely demented at very little detectable amyloids, while people with large amounts of amyloids had little to no mental deficit. Ouch! In the late 90s, I postulated that for a brain to function properly, it must have an unimpeded oxygen supply. Indeed, BERISLAV SLOKOVIC of the University Rochester Medical Center agreed, who claimed in 2008 that blood circulation plays a key role in the disease. I went one step further and concluded that where there is vascular plaque, the circulation is impeded. The cause of vascular plaque can be traced to hyperlipidemia, an excess of cholesterol. Indeed, if you look at it more closely, you will find that vascular plaque impedes, if not blocks, the serum flow and reduces, naturally, the oxygen supply to the brain. It follows that rather than targeting amyloids, the first target has to be the vascular plaque. And that is what I tried to achieve from the onset, back in the mid to late 90s. In the beginning we had some problems because the known drugs, also known as the reverse cholesterol transport triggers, didn't work so good. Why? Because, as I said, where the serum flow is impeded, the efficiency of the drug is reduced, hampered. Well, eventually we found the solution, a dramatic restoration of the healthy cholesterol levels in a few weeks. We filed several patent applications which cover an improved formulation for the prevention and treatment of dementia, Alzheimer's disease and hyperlipidemia. Designed that pill for that purpose, found an Italian manufacturer capable to produce the drug, and are in the process of forming a new pharma corporation in Europe, X-PHARMA, that will engage in further research, design and marketing of drugs. It may be noteworthy that our ADT3 drug has a dual market. First, the dementia Alzheimer market, but also the equally large market of patients who suffer from catastrophic levels of excess cholesterol, high levels of LDL, the bad cholesterol, and low levels of HDL, the good cholesterol. Oh, there is a reason why we want to start in Europe testing ADT3. We want to make sure that the drug is absolutely safe before we introduce it into the US market. That may seem overly cautious, but the excessive damage awards granted to people who claim injury would jeopardize the mere existence of the company and do damage to our investors. And it is my job to protect our investors. So, if you are interested, leave a message. Oh, did I mention that I'm taking ADT3 for 3 years now?